additionally fungal infections. And so she's going to talk to us about fungal infections and the lung microbiome, especially the lung microbiome in the ICU. Alison?
Great. Thank you so much. Appreciate that really kind introduction. I'm really happy to have joined the Rutgers community and thrilled to be talking to you today and look forward to meeting more of you in person over the course of the upcoming years. So I am going to talk, I'm going to, as mentioned, I do a lot of research in HIV-associated lung disease and COPD, and also do lung microbiome work and have done some of that as well in the intensive care unit. So I'm going to sort of focus on some of the more translational clinical aspects of that today. And please, I know, I don't know how you all generally run this, but I am more than happy to be interrupted if there are questions or comments or things that aren't clear. And if you're electronically raising your hand and I don't see it, please just interrupt me. I'm much rather have this as a discussion. So I'm going to start off. Oh, if I can move my slides. There we go. I am going to start off with a case. This is a clinical scenario. This is a patient that we had in Pittsburgh. This is before COVID, a 32-year-old male who came in with H1N1 pneumonia. He was empirically started on vancomycin, cefepime, and also tamivir. His bronchoscopy and his pleural fluid grew out MRSA. So his empiric cefepime was stopped. The other antibiotics were continued. But despite that, his fevers persisted. So we're left with kind of a common clinical scenario. What are the next steps for this patient? So a couple of options. You could repeat cultures, but can hold any additional antibiotics until you get those results back. You could repeat the cultures and start empiric Zosyn, or you could do a fungal workup. So what was done for this patient was sending off more cultures and starting Zosyn empirically. Cultures were all negative, but the patient improved. So we see this all the time in the ICU and in the hospital. Patients we think may have a pneumonia, may have another infection on empiric antibiotics, don't get anything back on cultures, but the patient gets better. So we're left with the question of why. Did our antibiotics actually treat something that we don't know about, or were they really unnecessary and the patient would have gotten better anyway? So what I'm going to do for the rest of the time is talk about how microbiome analyses and sequencing may help us in these kind of scenarios in the intensive care unit and in other patients with infection. So what I want to do first is talk about what is the microbiome and the lung microbiome, give you some definitions and some explanations of the methods, and then talk about how this may be applied in the intensive care unit, how we might diagnose people using microbiome sequencing. We're seeing a lot of that already. I don't know what tests you all have here, but we're starting to have at Pittsburgh have more blood sequencing for organisms, more respiratory viral panels, but I think this is something that's only going to continue to increase. How outcomes of patients may be related to their microbiome communities, and how looking at the microbiome may lead us to new therapies for our patients. All right, so to start out with, a couple terms. The microbiota is the microbes that collectively inhabit any ecosystem. So be it in the soil or in the lung or the gut, it's all the bacteria, viruses, fungi that are in that sample. The microbiome is actually slightly different in its definition, and that's the totality of the body's microorganisms, their genomes, their molecular products, and the surrounding environmental conditions. So you can think of it as sort of like the whole community and its conditions. The distinction between these are not not terribly important, but just you'll hear the different terms. And I think it's important to understand these as we see more and more research coming out about the microbiome. And I think it's only going to become more clinically relevant. Generally, the microbiome is detected by nucleic acid amplification and sequencing, and I'll talk about that a bit more. This is actually, while we've seen a real explosion in the field of the microbiome, this is actually microbiology and the study of what microbes inhabit a community is old. This is started in the 1600s with Van Leeuwenhoek, who looked at the microbes in his mouth to try to catalog what was there. The problem with those kind of techniques is that most of the microbes that we have, you can't culture them using traditional methods. So you can detect many, many more organisms using sensitive nucleic acid sequencing to pick up the DNA or in some cases the RNA of the organisms that are there. And this development of next generation sequencing has really enabled the study of the microbiome. What I'm going to mostly talk about is the 16S rRNA sequencing, and this you'll see very commonly in many studies. And this is to detect bacteria, and it takes advantage of the fact that there's this gene called 16S rRNA that's present in all bacteria. It's essential for transcribing mRNA into amino acids and proteins. And it has both conserved and variable regions. So there are regions of the gene that are the same throughout all bacteria. And then there are regions that are variable. So you can use that. You can use the regions that are the same to do your sequencing so you can amplify the bacteria. And then using the variable regions, it helps you identify which particular bacteria are there. So this is a schematic of the steps in microbiome analysis. You sample your microbial community. For us, often we're doing bronchoscopies or endotracheal aspirates since we're interested in the lung. You extract the DNA, then you amplify and sequence the 16S gene. And then you take the sequences that you get and align them and group them together into what are called operational taxonomic units or OTUs. And these are sequences that are at least 97% similar to each other. And you can sort of roughly think of these as species, although sometimes it's hard to identify which is the particular species, but it tells you which types of and how many bacteria are in a given sample. And then there's many, many ways that these can be analyzed. Just going to give you a couple of basic terms that are commonly used. You'll see the terms alpha and beta diversity quite commonly in the literature, and I'll be talking about that as well. Alpha diversity is how variable is my community. So if you look, how many different bacteria are there, and are they relatively even in their distribution? So you can think of it as this coral reef here. That's a very diverse community. You can see lots of different brightly colored fishes, and they're all sort of relatively even, as opposed to the one with all the dead fishes, right? That's a non-diverse, low alpha diversity community because it's just dominated by one particular species. And you can think of that, generally that's associated with disease. So things like C. diff have very low alpha diversity. Beta diversity is when you're looking at differences in all the bacteria or organisms in different communities or in different samples. So beta diversity here on the right, each of these dots is the microbial structure of a certain sample from a patient. And as you can look at the groupings and pretty quickly see by eye that healthy patients are different from those with a disease. And you can do this for whatever sample, whatever condition you want. So it gives you sort of a way of visualizing how similar or different are the communities. And as I mentioned, the study of the microbiome is really only increasing. We now know it contributes to many diseases. It's really hard to find any condition where there's not been some study linking it to the microbiome. Most of the microbes live in the intestinal tract. So there's been lots and lots of research into the gut microbiome and how it influences not just the gut, but because the gut has lots of effects on the immune system systemically, lots of metabolic products, how it may influence systemic disease. The lung microbiome, which is more my interest as a pulmonologist, is really far behind the gut field. People for many years thought that there was no lung microbiome, that the lung was sterile. If you think about it, that doesn't make a whole lot of sense. Well, clearly it doesn't have all the bacteria that the gut has. I mean, the lung is in constant communication with the environment. It's in communication with the upper respiratory tract, the mouth, which has lots of microbes in it, the nasal passages, and we breathe in and out. So it did not make any sense that there would be no microbes in the lung. And what we've come to realize over the past 10 years or so is that there is a lung microbiome. It's very low level. There aren't tons of organisms there, but they are there, and they are there, can be there transiently. And the normal lung microbiome is primarily derived from oral bacteria. So everybody has at least some sort of microaspiration, can get worse in patients with disease. But throughout the day, some of the oral microbes do get down into the lung, and that's primarily what we see in the healthy lung microbiome. All right, so let's go back to our case. And we're interested in why this patient got better. And we're fortunate that he was part of a study that we have been running at Pitt that was led by Georgios Kitsios, who's shown here. And this was a study where in our intensive care unit patients, we collected longitudinal oral swabs, tracheal aspirates, stool and rectal swabs, and blood samples. And the similarity between Dr. Kitsios and the representative ICU patient here is purely coincidental. All right. So this patient was part of that study. So we had samples that we could perform microbiome sequencing for, and we did the bacterial sequencing. So again, I'm primarily focusing on the 16S sequencing, which is for bacteria. We can also do similar things for fungi and for viruses, although it gets a little bit more complicated. But we did 16S sequencing of the endotracheal aspirate from this patient and showed a predominance of Fusobacterium, an organism that's hard to grow, and an organism that's sensitive to Zosyn. So we had an explanation of why this patient got better, actually did respond to the Zosyn, and that was the right thing to do, even though we didn't actually know that at the time. So, this is really a common conundrum in the ICU, right? Our patients come in and they're critically ill, and we know that less time to effective antibiotics improves outcomes. That's been clearly shown. But our cultures take several days to get results, and they're insensitive. So in the meantime, we give empiric antibiotics. These have a lot of adverse effects, cost money. You can get things like C. diff, secondary infections, and it leads to the development of antibiotic resistance. So we wanted to ask the question, could we use sequencing to improve this traditional diagnostic scheme where we're taking samples, waiting several days for the cultures, and in the meantime, giving patients broad spectrum antibiotics? So this was a preliminary study that Georgios did with this group of patients. And we took 56 mechanically ventilated patients, had endotracheal aspirates, which, although they're called endotracheal, it's actually we go through the endotracheal tube and it's fairly far down in the lung. It's not actually in the tube. So it's it's not a bronchoscopy, but it's a reasonable representation of what's in the lung. We did 16S sequencing. And 12 of the patients had negative or had positive cultures by traditional methods, and 44 had negative cultures. So we used our sequencing to look at the community structure and to see if we could identify pathogens. And this is what we found. So this is the alpha diversity and the beta diversity of these samples. And for those patients who had negative cultures, they had a higher overall alpha diversity, meaning their microbial communities were more diverse, had more organisms, compared to the culture positive, where there was a low alpha diversity. And that sort of makes sense, right? If you had an organism or pathogen grow out on culture, that's probably the dominant thing, and there's not a lot of other bacteria in that sample. When you look at the beta diversity, so comparing the communities in culture positive versus culture negative, you can see that they also group distinctly. But what was interesting in this is if you look at this group of patients who were culture negative but had a low alpha diversity, they look fairly similar to those patients who did grow an organism. And when we looked, these patients had more inflammation. So they had greater systemic IL-6, they had higher TNF alpha in these patients who were culture negative but had low diversity samples. So it raised the question, are we missing pneumonia? And we looked at what we had found by the sequencing in these individuals. And on the left are those culture positive patients. And the red, each bar here is an endotracheal aspirate with the proportion of the type of bacteria that we detected. So red means pathogen. So for example, this one on the far left has 100% of pathogen in the sample. Blue is oral bacteria, and then gray are just various other things that we found. So you can see all the culture positive ones look fairly similar, have a high predominance of pathogen. When we look at the culture negative, if you look on the left, those ones that had a low alpha diversity also had a high proportion of detection of what we would consider respiratory pathogens, so Pseudomonas, Staph, Enterobacter. So it made us wonder, perhaps that these are patients, if we had had this sequencing, would not have gotten would have gotten antibiotics, but we could have stopped it in these other patients who were culture negative and had predominance of oral bacteria, which, as I told you, is sort of what we expect in the normal lung. So we then ran a clinical simulation to see if we had used that data to make antibiotic decisions, what would have happened. And we found that there would have been a 50% reduction in empiric antibiotics with 23% of the total courses avoided. So a pretty big impact in this patient population. I'm going to give you one more example to show that in many situations culture may be or sequencing may be superior to culture. So this is a patient who came into our ICU who had septic thrombophlebitis, grew out in the blood fusobacterium. Patient went into ARDS with hypoxemia, fevers, and shock, and developed pneumothorax and pleural effusion. And interestingly, a week later, the pleural fluid grew out mycoplasma. So kind of an unexpected result. And nothing, not an organism that he was empirically treated for. We had multiple samples from this patient to do sequencing, and we were able to detect the mycoplasma far before our cultures were able to. So if you see, this is this is sequencing from the initial time when he came in. The majority of the bacteria detected were the blue fusobacterium, but almost a quarter of the bacteria detected in the endotracheal aspirate was actually mycoplasma. And you can see over time, the percentage of mycoplasma increased until it became the dominant organism. So again, suggests that maybe if we had had this information earlier on in the course, we could have tailored the antibiotics better. Now, the problem with that, though, is the 16S sequencing is difficult to apply in clinical practice. It's slow. It takes a while to run. There are biases from the PCR, the amplification steps that we do. We get limited resolution. It's hard to get down to this species specificity. We don't get information about antibiotic resistance, and it's limited to only bacteria. So with more modern sequencing or more complete sequencing, we're able to, first of all, do this faster and more completely. So this takes advantage of metagenomics, and that's instead of just looking at the 16S in bacteria, it looks at all the genetic material in a sample, and it sequences the entire genome. And that gives you better resolution, better identification, and you can sequence things beyond bacteria. Actually, this is the machine that it's done on. It's a nanopore machine, and it's called the Minion or Minion. And it's very small. It's smaller than your iPhone, and it can do this sequencing very quickly. So we then did a proof of concept study using this nanopore to see if we could identify pathogens quickly in ICU patients. And this is just a summary of what we found. We were able to do this in 6 hours, and we found concordance between the dominant pathogen in our samples, that's shown in red, and what was detected by culture and what we could detect by this sequencing. And then there were a number of culture negative patients where we detected primarily Candida in the samples. So not quite ready to be used in the ICU, but I think will be in the next few years. And again, this raises the question, can we reimagine how we diagnose pneumonia? Instead of using these cultures that take a long time and expose our patients to antibiotics over time, can we use next generation sequencing? And I've talked primarily about respiratory specimens, but this can be done in blood as well. And can this give us a culture independent diagnosis that will improve patient care? All right, I am going to shift a little bit and talk about the impact of these microbes on our patients. So this is another common scenario that we see in the ICU. This was a 53-year-old female who was generally healthy, who came in with fevers, dyspnea, constitutional symptoms, was diagnosed with H. flu pneumonia. She rapidly evolved, was admitted to the ICU with acute respiratory distress syndrome and multi-organ dysfunction. And the question is why, right? We see lots of patients who come in with pneumonia, they get some antibiotics, they go home. Why do some people get so much worse and wind up in the ICU? Just a little bit of background on acute respiratory distress syndrome, or ARDS. It's probably familiar to everyone, but it is a heterogeneous clinical syndrome. It's defined by hypoxemia, radiographic edema, that's diffuse and bilateral, with a risk factor for lung injury. Pre-COVID, it was responsible for about 10% of ICU admissions. Has a high mortality of 30 to 40%, and long-term morbidity in people who survive. And despite many years of study, there is no broadly efficacious pharmacological therapy. And there's been multiple mechanisms as shown on this figure on the right, trying to understand the immune response, the inflammatory pathways, and the lung damage that occurs in order to try to intervene. So there's been a number of studies, first done by Carolyn Colfie's group in San Francisco, and some studies that our group has done, as well as others, both nationally and internationally, trying to look at why patients with ARDS are different and trying to understand the causes of it and how we might treat patients differently. So these studies took unselected ARDS populations and then did latent class analysis. And this identified 2 different ARDS subphenotypes. So one was hyperinflammatory and the other is hypoinflammatory. And this is based on underlying biological differences. So hyperinflammatory, as the name implies, increases in cytokines, higher tissue injury, metabolic acidosis, and organ dysfunction. Whereas the hypoinflammatory patients had a much lower levels of all these markers. And why does this matter? These patients seem to have different outcomes. So the hyperinflammatory group has lower survival over time. And there may also be differences in the treatment response. You can imagine if you have lots more inflammation, you may respond differently to an anti-inflammatory than a patient who has ARDS, but does not have that inflammation. So maybe one of the reasons why when you look at these studies of therapies in ARDS, you don't see an effect because we're not looking at the proper subgroups of patients. So based on these classifications, we thought, well, can the microbiome help us understand the differences in the host response and outcomes of these hyper and hypoinflammatory patients? Might the underlying microbiome be something that's stimulating or worsening the inflammation that occurs in the hyperinflammatory patients? And maybe those patients who are hypoinflammatory have a different body microbiome. So we looked at about 300 mechanically ventilated patients with acute respiratory failure. We did oropharyngeal swabs, endotracheal aspirates, and blood samples. We did the 16S sequencing for bacteria on the oral swabs and the endotracheal aspirates, and then did a panel looking at host response biomarkers in the blood. And then we used these Dirichlet multinomial models, DMM, or a cluster analysis to say, can we group these patients based on their microbiome? To approach it agnostically and say, if we know nothing else about the patients and we just want to see, are there groups that are similar based on their microbial structures? And then we looked at the relationship of that to outcomes. So 30-day mortality and liberation from mechanical ventilation. And what we found was really interesting. I'm going to present primarily the lung data. The mouth data was pretty similar. Low microbial diversity in the lung was associated with worse survival. And you can see this is the alpha diversity, with survivors being higher in their diversity than non-survivors. And the beta diversity on the left, you can see the communities look different. Interestingly, this low diversity was also associated with secondary infections. So those patients who went on to develop ventilator-associated pneumonia or other infections had lower diversity in the lung at the beginning of their ICU stay compared to those who did not. We then went and did the clustering analysis that I mentioned. And we found, based on the bacterial communities in the lung, we found 3 distinct clusters that we creatively named cluster 1, 2, and 3. And these had different microbial compositions. So cluster 3, which you can see has the highest alpha diversity, looked pretty much like the mouth, right? So we see things like Veillonella, Prevotella, some strep in blue, and then very little pathogen like staph shown in red. And so the size of these bubbles represents how much of each bacteria was in these communities. Cluster 1, which had, you can see sort of in the middle of alpha diversity between cluster 2 and 3, had kind of more of a mix, had a fair amount of mouth bacteria, but also had a little bit more pathogens, so staph and things like that, in the lung. And cluster 2 was the worst. You can see more red there. It had more staph, Pseudomonas, Enterobacter, and much less oral bacteria. And interestingly, these were highly associated with outcomes. So survival was the worst in those cluster 2 patients that had the most pathogens, and was the best in cluster 3, where the lung looked more like the mouth microbiome. Similarly, liberation from mechanical ventilation, patients in cluster 3 were more likely to be extubated than patients in cluster 2. We then went back to that hyperinflammatory scheme and found that indeed cluster 2 was more likely to be hyperinflammatory and to have higher inflammation both in their lungs and in their blood. And what was interesting was it wasn't just patients with pneumonia, right? So going back to the data I showed you in the first part of the talk, these low diversity hyperinflammatory patients where we found pathogens on sequencing didn't necessarily have either clinical pneumonia or were culture positive. So it gave us more information about what was going on with the patient than we could see by traditional examination. And these findings have now been replicated in a very large ICU meta-analysis. So this was a study that came out 2 years ago, where they looked at 17 studies in greater than 1,000 ICU patients and over 300 healthy controls. And they harmonized the 16S data to compare and come up with analyses for all of these patients. And what they found was very similar to what we found. Patients with ARDS, patients with hospital-acquired pneumonia or prolonged ventilation had distinct microbial signatures, and those were associated with depletion of the oral microbes in the lung. And enrichment with pathogens such as Pseudomonas and staph, particularly which were associated with the development of pneumonia. So, now this has been replicated in many studies, and I think we're really seeing that there is an association with outcome with the underlying microbial communities. And I would say that this is when the patient first comes into the ICU. So this can predict events throughout the hospital stay. Now, we were also interested in what is happening over that hospital stay. We do a lot of things in the ICU that mess up the patient's microbiome. We give antibiotics. We used to cleanse the mouth. Patients have tubes and lines everywhere. We feed them at all different times with tube feeds. Lots of things going on that could impact microbial diversity. And so this is data showing longitudinal samples from the mouth, the lung, and the gut. And this is the alpha diversity. And you can see the first bar, this is healthy control patients. And then this is baseline in the ICU, middle period of the ICU stay, and later in the ICU stay, which was 2 weeks after the patient was admitted. And you can see that in all of these compartments, the diversity of the microbiome decreases over time as patients are in the ICU. And again, this decreased diversity is associated with a poor outcome. So those patients who were low diversity and persisted that way had the worst outcome in terms of survival. They were least likely to survive. The patients who had some improvement in their diversity over time seemed to fare better. So I think this data taken together helps us to rethink some of this paradigm of the heterogeneity of ARDS patients and where these subphenotypes may be coming from. So perhaps there are underlying changes in the microbiome. And I showed you primarily the lung, but again, the mouth seems to be related as well. Less so the gut, although there have been some studies that have shown differences in the gut microbiome in ICU patients that are linked to outcomes. And that perhaps this is part of what's driving this inflammation and the differences in outcomes and the differences in treatment. So how does this translate? I think it really raises the question of intervention, right? So this some of our findings may be in association and not actually causative, right? So as I said, we do a lot of things that mess up the microbiome in the ICU. Patients may be coming in with changes because of their infection or whatever is going on. So maybe this is just all an epiphenomenon. But I think the strength of the associations, the relationship with inflammation and the relationships with survival suggest that perhaps there is an underlying mechanistic link to the microbiome to outcomes. And that may be some place where we can intervene to help improve the outcomes of our patients who get so sick with ARDS, or perhaps to prevent ARDS when the patient I showed you comes in with their H flu pneumonia. Can we do some sort of microbiome therapeutic to prevent the development of lung injury and poor outcomes? So many ways that we might be able to approach this, and we're doing several studies trying to sort this out, but it raises the question of how we can intervene. So, for example, oral hygiene, right? Maybe having good bacteria in your mouth is better for your lung, and that will decrease your inflammation and your severity of illness. Should we be using mouthwash? Should we be targeting specific bugs with antibiotics? Should we be using probiotics? Would that help if we can impact the mouth, the lung, the gut with giving particular bacteria to influence the communities? Should we be transplanting bacterial communities? We do fecal transplants for C. diff. Should we be doing lung microbiome transplants or oral microbiome transplants to impact the communities there and help improve patient outcomes? And there are lots of additional studies that are going on to look at this more. And also I should mention, should we be targeting the inflammation instead? Should we be doing particular anti-inflammatories? We're starting to see more evidence of that in COVID and in other pneumonia, that perhaps targeted anti-inflammatory strategies may help what pathways are being stimulated by particular microbes. And I'm just going to finish with this concept of the oral bacteria and perhaps that they are helpful. So I showed you several pieces of data that suggest that patients who do better have more oral bacteria in their lungs. And they have sort of the normal oral bacteria that we see in healthy patients. So our cluster 3 patients who did well had primarily oral bacteria. Our culture negative patients who had a predominance of oral bacteria didn't seem to have a pathogen and didn't seem to have much inflammation. So, is this just an association, or is this something that might actually impact outcomes? And so I'm going to show you a really cool study that was done by Benjamin Wu and Leo Segal's group at NYU. And this was done in a mouse model of strep pneumonia. And what they did was they took sort of a normal oral microbiome community, so strep, villanella, and prevotella. They gave it to the mice. They put it down in the lungs as if the mice were aspirating this, right? They then infected the mice with strep pneumonia and interestingly found that the mice that had received the oral microbes had lower strep numbers cultured out of the lung an infection, and they had better survival. So you can see in purple down here, these are the mice that got the oral bacteria. And to both a sublethal challenge and a lethal challenge with strep, they had much, much better survival than those animals that just got a control aspiration. And if you look, the numbers that they got out for culturing strep were much lower in those animals that got this oral aspiration. And what was really interesting about it was that these oral bugs were actually gone. They were cleared out of the lung and couldn't be detected prior to them inoculating the mice with strep. So there was some effect that these oral microbes had on the lung immune environment that helped them get better from pneumonia. So is this something that we can do in people? Should people be taking some sort of oral probiotic? Should we be giving that to people in the ICU or putting it down into the lungs to help protect them from secondary pneumonia? I think these are all really interesting questions to try to answer in the next few years. So in conclusion, I hope I've shown you that microbiome techniques offer potential for improved sensitivity and rapidity for infection diagnosis. And while I focused on the ICU, I think this is true for many infections, not just pneumonia, not just ICU patients. Disordered microbial communities in the lung relate to important ICU outcomes. Oral bacterial communities in the lung may be beneficial, and we may be able to, in the future, base novel treatment strategies on the microbiome. So I just wanted to acknowledge all the people from my lab who did this work and the people who were involved in the ICU study, collecting the samples, and then particularly the ICU patients and their families, and our funding from NIH. So thank you very much, and I will be happy to take any questions.
Thank you so much, Alison. That was so interesting and wonderful. I actually have a huge number of questions, but I don't want to hog the uh, question answer sessions. But I do want to start with a question. And I think, when you showed the low alpha diversity culture negative group, uh, I was just curious to see. I mean, presumably their survival is lower than the culture positive group because we didn't have culture positivity and we didn't understand treatment unless empiric antibiotics hit the bug that was causing that culture negativity. But my question is, what's the reason for that culture negativity? What's the difference between the culture positive and culture negative in the low alpha diversity group? Is there is just lower pathogenic burden or?
Yeah, it's a combination. I mean, so we know most microbes can't be cultured, right? And so
Right.
like that fusobacterium case I showed you, that's that's a bug that's often very hard to culture. So I think it's a combination of lower levels of the organism. All these patients, I should mention, all these patients are on antibiotics, right? Because they come into the ICU and they're sick and they get put on antibiotics. And we see all this despite that, right? So all our analyses are adjusted for that. But they are on antibiotics, so their cultures may be less likely to grow, maybe a lower organism burden, or it may be the particular type of organism. And it may just
I
be
was
something like at low levels, and then we see it increase later on.
right, but have you done like a cluster analysis? Is there a hyperinflammatory or hypoinflammatory state in these
Yeah,
patients?
so the low diversity group with pathogens by sequencing, um, but culture negative, are hyperinflammatory.
Okay. So it may be cause and effect rather than, I mean, we can't
Yeah,
tell chicken or egg, right? Is it hyperinflammatory because of the particular organisms, or is it those organisms aren't being grown
right,
because of the
right.
hyperinflammatory state?
Yeah,
Right.
yeah.
All right. Okay. I'll come back to more questions if we have time, but I think Dr. Clark, our chair of medicine, has a question.
Thanks so much, Allison. Super fascinating talk. Really, really interesting. I want to ask you a question about in the ARDS group when you were, talking about diversity and so forth. In individual patients, right? You found very statistically significant or neat patterns in groups, but in individual patients, we often need some sort of cutoff, and there's a lot of overlap right in there. Do you see a way forward, or what would be the next steps to sort of say this is the patient, who has the poorer prognosis, is in group one, or however you want to classify them, because all the graphs you showed really had a lot of overlap, right? And so
Yeah,
how would you apply that ultimately to individual patients, or have you thought about that?
yeah. Because obviously the goal at the end of all of this is to have something that when a patient comes in, we could say, okay, this is a high-risk patient, and we're going to manipulate their microbiome this way. There are a couple ways that could be done. One is using an abundance cutoff. So if we see over a certain amount of a pathogen or of a disordered community, that we can say that this is a patient that is at risk. We also have generated a dysbiosis index. and I didn't show that data today, but using that data, you can generate an overall index that summarizes an individual patient's data, and that has really pretty good predictive value, as good as anything else. And then it hinges on being able to get that sequencing quickly, right? And so I don't know, maybe if we can get the sequencing within 6 hours and know what's there, just treating with appropriate antibiotics or stopping antibiotics may be sufficient. We don't need to do anything else to help improve, outcomes, or maybe we need to, give them an oral bacterial transplant. The challenge will be, I think, getting physicians to stop antibiotics based on this kind of data, right? Because everybody always feels like my patient's really sick and I need to be giving antibiotics. And so we've done some work, trying to the study looking at the empiric outcomes or the theoretical outcomes with empiric antibiotics. It may help, but it'll probably have to come down to some sort of trial where patients are assigned to having this or not, and trying to convince physicians that it's okay to stop your antibiotics based on this.
Or as you've implied, redoing or changing some of what's now become norm, right? Hemocleanse or mouthwashes and some of that stuff, right? We're changing that paradigm for some patients, right? Sounds like maybe interesting.
Yeah,
Anyway.
and maybe not everyone, right? It will, I think, end up in very personalized medicine.
All right, so we have
very nice. Thank you.
We have a couple of questions in chat. Dr. Butner, Chief of Endocrine, asks, what if the altered microbiome is simply an epiphenomenon and not the driver in some cases of ARDS, reflecting altered immune responses and overall changed organismal health? Some antibiotics seem to alter immune responses independent of their ability to kill bacteria in the lung. If that is true, maybe there are limits to how to be guided by the lung microbiome when selecting antibiotic therapy.
Yeah, so I think that's a really good question. And I think there's two parts. There's one, guiding therapy based on sort of this idea of it stimulating inflammation and impacting outcome. And then there's two, what we were just talking about, the using it for diagnosis of pathogens, right? And I think it's pretty clear that for that scenario, that this sequencing is going to be better. I mean, we're already using it a lot, not necessarily like the nanopore metagenomic sequencing, but there's a lot currently in standard of care. But yes, in terms of the relationship with outcomes and whether it's a driver of ARDS or not, is much harder to untangle, right? There's antibiotics that have a myriad of effects. Lots of other things going on with these patients. And so that's where I think animal models help a lot. So like that study with giving the oral bacteria to the mice and improving outcome from strep pneumonia, right? Suggests that there's something that's driving, not necessarily in every patient, but perhaps in some. Our group's doing other studies looking at translocation of these bacteria. We have a lot of data I didn't show today that translocation from the lung or the gut of bacteria, fungi, viruses, if you can detect it in the blood, it's much worse, and is associated with more inflammation in these outcomes. So can you perhaps make the gut or the lung less leaky, and not have the microbes or their products leak out? But I think it's going to come down to to doing animal models and then doing some therapeutic trials, because that's really the only way you're going to get at cause and effect.
Actually, that's a follow-up question that I actually it's a good question, and Dr. Azim, who's our infectious disease faculty member, has a question, but I'd like to ask my question before this question, which is, we know aspiration pneumonia predisposes to ARDS. So if you're telling us that oral bacteria are good, right, and the aspiration event, however, predisposes to ARDS, how do we kind of reconcile the two? Is it just inflammation and hyperinflammatory state, no matter what the trigger is, which potentially could be, right? Any, I guess I'm asking you to speculate on that. And then I'll go on to Dr. Azim's question because it's related.
Yeah, so I think it's part of it is what you aspirate. So if you aspirate like good oral bacteria, then maybe you're better off. If you have disordered oral microbial communities, which we see and which is also connected to outcomes, then maybe that's worse. But then, if you have a huge aspiration of gastric contents that causes a sterile inflammation, and then, secondary infection, I think that's a different phenomenon. We've actually looked at using sequencing to look at presumed aspiration versus not, and we don't see actually a whole lot of
Oral.
differences. Yeah. In terms of what's down there.
So his question is, despite the ATS IDSA guidelines recommending against anaerobic coverage for aspiration pneumonia, it's still a very common practice that will likely persist for years. Do you worry about the continued use of anaerobic coverage, for example, pip/tazo, for aspiration pneumonia, knowing that we are potentially wiping out good oral bacteria?
Yes, absolutely. I mean, I think that's where the sequencing could be helpful. There's a group in Michigan, Bob Dixon's group, that has shown worse ICU outcomes, particularly with anaerobic coverage.
Okay, and I have one last question because we still have some time. You mentioned tube feeds, which I find fascinating, and I'm more interested in the circadian timing of tube feeds and the outcomes in ICU because that's never been studied, and we seem to feed people 24/7. That's not normally, and that's not normal. Has there been any data on changes in the lung microbiome with 24-hour versus bolus feeds versus, regular eating?
Yeah, no, I think it's a really interesting topic because the microbes are circadian too, right? And their
Right.
function, they change their gene expression, and they're not used to being fed around the clock with these artificial things. But I don't know of data that's looked at the relationship of timing with the lung microbiome. It would be interesting.
Yeah, I think a project for one of our junior faculty or fellows.
There you go.
Yeah, I get them interested in that. Are there any other questions? Let me see. In the chat, I didn't see any others. Make sure I'm not missing anybody raising their hand here. There are quite a few. I don't see any raised hands. Well, in that case, thank you so much, Alison. It was wonderful, and thanks again for the presentation.
Thank you.
Bye.